Okay, yeah, so welcome to this month's Myeloid Network seminar. We'd like to thank our sponsors, especially the Moores Cancer Center and Adam Prater, who support this program. We also would like to thank AstraZeneca and Charisma for their financial support. We have one announcement before I introduce our speaker today, and that's the slide that's on your screen now. The Myeloid Network has partnered with SITC again this year to run a session at the SITC annual meeting, so I hope that you'll join us there. And so I'm delighted to introduce our September speaker, Dr. Christine Moussion. She has extensive background in immunology, having earned her Ph.D. at the University of Toulouse in France, where she studied the role of dendritic cells in regulating lymphocyte migration from the blood to the lymph nodes in Jean-Philippe Girard's lab at the Institute of Pharmacology and Structural Biology. Following this, she completed a postdoctoral fellowship at the Institute of Science and Technology Austria in Vienna and Michael Six's group, investigating the migration of dendritic cells from peripheral tissue to draining lymph nodes, being lymphatic circulation. In 2017, Dr. Moussion joined Genentech in San Francisco, California as a group leader in the Cancer Immunology department. Her team focused on bridging basic science with drug discovery, deciphering novel cellular and molecular mechanisms that restrict leukocyte recruitment and anti-tumor immune responses, spanning from mouse models to human pathology, and utilizing cutting-edge, high-throughput in vivo technologies, which was published in Nature with Dr. Moussion as the senior author, along with genetic and pharmacological approaches. Her group has uncovered novel therapeutic modalities to improve immunotherapy outcomes in solid tumors. In 2024, Dr. Moussion became the therapeutic strategy lead for tumor microenvironment in the Cancer Immunology Discovery Department at Genentech. Dr. Moussion, we are very excited to have you here. Thank you so much. And so I will turn it over to you.
Thank you so much, Jen, for this kind introduction. It's my great pleasure to be part of this Myeloid series. And I'm also, thank you for the invitation, and I'm also very looking forward to the Myeloid session at SITC. So please join there. I'm going to share my screen. Okay. So I will present the work that have been developed in my lab for the past years. And this talk is in two parts. The first part, I will talk about the STAMP technology that we have developed to study the tumor microenvironment and the myeloid cells. And the second part, I will talk about a new clinical candidate molecules that has been developed at Genentech to leverage dendritic cells and monocytes for cancer immunity. So this talk is on behalf of many collaborators. Genentech is a big company, so it's a big collaborative environment. And as I mentioned, we are really fan of live imaging of immune cells in tumor and looking at dynamics, as you can see here on this movie. So a disclaimer, I'm employee at Genentech and on Roche stocks. First, a quick definition of what we call tumor microenvironment. So we know that solid tumor are dynamic population of cancer, immune, and stromal cells that vary in function and in frequency, and that also vary in time. So this environment is dynamic. Many people focus on cytotoxic T cells in the tumor microenvironment, but we should really keep in mind that all those other cells are there and really interacting with the T cells and controlling their function. Those are the case of the myeloid cells, the stromal, lymphatic, blood vessel innervation, and also soluble factor that are controlling the function of the immune cells. Based on anatomopathology and immunohistochemistry in patients, solid tumor can be classified as three tumor immune phenotypes based on CD8 T cell location: immune inflamed tumor where CD8 T cells infiltrate the core of the tumor, immune excluded tumor when T cells are stuck outside of the tumor nest in the tumor stroma, and immune desert tumor where CD8 T cells are not recruited in tumor. We also know that those immune phenotypes are important because they are associated with clinical responses to immunotherapy, the immune inflamed phenotype being a prognostic and a predictive biomarker of response. So, however, the immune desert and the immune excluded tumor here are the highest unmet medical needs, which is a majority of tumor across indication. And our goal in the cancer immunology department is to convert this tumor from immune desert to immune inflamed. So although those immune phenotypes are well characterized in patients, we realize that we know little about their development, their heterogeneity, or their dynamic. And we thought that there is a missing link between the clinic and the preclinical model. So we decided to develop a new technology to be able to image this tumor microenvironment in vivo by high throughput. And this is a teamwork between wet lab scientists and computational scientists, which is very important in my group. We always go back and forth between computational and wet lab, but also between mouse and human. This is how we make sure that our biology translates in between species. So this new technology, we call it a screen tumor array by microporation. And this is a novel, non-invasive, high-throughput imaging technology that explores the spatial organization and function of immune cells in an anesthetized mouse. So what we do is we use a laser that is creating an array of pores on the ear on the animals. And then this is just a physical way to open the barrier, and we implant tumor cells that are expressing a GFP reporter here into these pores. And so we implant those tumor nodules 50 to 90 micron below the epidermis. So this is very superficial, and that allows us to perform live imaging daily up to a month. So we take images, and then what we do is we measure the area of those tumor nodules by looking at the green fluorescent protein. And then we had to spend a long time on, this was actually the most challenging part of developing an image processing and analysis pipeline that is able to segment and quantify those tumors over time, because you are now looking at several thousand of tumors in the same experiment, and this is pretty laborious if you have to do it manually. So what we have done is to develop this automated pipeline image analysis, and how that works is that you have here this stack and time that is composed of two channels, the green channel for the tumor cells, and then the red channel here are the immune cells, the T cells. And you can see those are different spatial distribution around the different tumor nodules. So the first step is to perform tumor segmentation, where we basically detect the tumor area versus the normal adjacent. And then using a different algorithm of segmentation, we basically detect the different tumor nodules. And the second part is to track those tumor nodules over time. So now we have to tell the software that this tumor is the same as this tumor, so we link the tumor over time together. And each time we have a small module of manual curation to make sure that there is no mistake. And then for what we do for each tumor nodule, we are now going to look at the spatial distribution of immune cells. So we perform a radial analysis where we basically draw a radius that we spin around is 360 degrees, and then we represent the average fluorescence of immune cell channel along this radius. So I will show an example later. Basically, what we get from that is a 2D map of the immune phenotype of the different tumor nodule between excluded, inflamed, and desert. And then we developed a Shiny app to explore those data in high throughput, where we can really interrogate experiments of several thousand of tumors over time. So which type of parameter are we quantifying? We are looking at tumor growth curve, as I mentioned, over time. And this is an array of 100 tumors, where one line is one tumor growth, and you can see that some tumors are growing and some tumors are getting rejected over time. We also measure what we call tumor survival probability, that is represented as a Kaplan-Meier with confidence interval, where we represent the survival probability of a tumor. So when the line is flat line on the tumor are growing, and when the line is dropping, you see here you can observe a 30% of rejection. And then the last parameter we are measuring, I mentioned, is a spatial distribution of T cells for each tumor nodule. So we perform this radial analysis, and we measure and we represent the average fluorescence intensity of both channels, tumor cells in blue and T cells in red, along this diameter here. This is the center of the tumor, and you can see that when both fluorescents are overlapping, this tumor is called immune inflamed. When the fluorescents are mutually exclusive, this tumor is called immune excluded. And then we don't only classify the tumor as inflamed, excluded, or desert, but we also measure it in a continuous way, and we calculate what we call the infiltration ratio, which is the T cell abundance outside divided by the T cell abundance in the core of the tumor. So that's more or less the parameter we are extracting for several thousand of tumor over time. And then we can plot those different parameters here using the app. So just to summarize what this model allowed us to do, this model is high throughput. It's also longitudinal. We can follow tumor over time. We can study microenvironment, and we can image at high resolution each tumor nodule. The advantage over subcutaneous tumor is that it allows you to follow tumor rejection. So you will see over time the tumors that are growing and then getting rejected. So you can really learn from an efficient rejection at the opposite of a subcutaneous tumor where you really need to harvest the tumor at a certain time point. That means the tumor is a failed rejection. It has to keep growing for you to be able to sample it. So what did we learn with that system? So it's just a technology, but what we are mostly interested in is to learn some biology out of it. And so what we have looked at first is this spontaneous local tumor rejection. So we realized that when we implant this tumor array in wild-type mice, you can see in a single tumor array, some tumor nodules are growing and some tumor monodules are getting rejected. If we implant that in a RAC2 deficient mice, 100% of tumor are growing. So I remind you that these tumors are implanted in the single animal, so with the same T cell repertoire across the organism and the same blood circulation. And also what we have done before implanting those tumors, we have performed a single cell clone to make sure that all the tumors that are deposited in those pores are genetically identical. So those tumors are genetically identical, however, some are going to grow and some are going to be rejected, and they are located 1 millimeter apart. So this was very intriguing to us. And as I mentioned, another way to represent it is in rag mice. You see here a flat line in the Kaplan-Meier representation, where in the wild-type animal, more or less 30% of tumors are spontaneously rejected. So the first question we are asking is that at all T-cell dependent? And is it antigen dependent? So the answer, we have done many adoptive transfer. We have transferred bystander T cells versus antigen-specific T cells. We have performed CD8 depletion. We have also performed a transfer of T cells from vaccinated mice that have been vaccinated with tumor neoantigen, constitutive antigen. And the answer is that this rejection is CD4 mediated and CD8 mediated, and this is antigen specific. Rejection. So it seems that there is an heterogeneity of tumor fate that is locally controlled by CD8 T cell responses. So what is controlling this heterogeneity of tumor fate? So what we have done is then to perform live imaging of the tumor array and looking at both tumor cells and T cells. And for that, we implanted the GFP tumor in a rag mice and then reconstituted the mouse with TD tomato T cells. And what we see across the tumor array is an heterogeneity of tumor immune phenotype. So you can see here some tumors among this array are very inflamed, with the green and the red overlapping. Some tumors that are 1 millimeter diameter here are pretty excluded, with the T cells that are stuck outside of the tumor core. Some tumors are ignored, so they're very desert, they don't recruit any T cells. And then we have defined a fourth phenotype that we call rejected, where the GFP is disappearing over time, but the T cells stay in the scar of the rejection. And this cloud of T cells will stay there for several weeks, and sometimes months. So we clearly be able to reproduce those different tumor immune phenotypes in vivo in mice, and being able to image them over time allow us to define those four phenotypes. So the first question we have been asking is this model relevant at all for human pathology? And it is important for us because if we start testing immunotherapy or immunotherapeutics, we need to figure out that this biology is reproducible. So the first thing we have done is perform punch biopsies of those different nodules and perform a bulk RNA-seq. And we see that in the principal component space, those tumors cluster together per immune phenotype. So the immune-inflamed tumor cluster together and are enriched for interferon alpha and interferon responses, interferon gamma responses, and allograft rejection gene signature. The immune-excluded here are enriched in hypoxia, angiogenesis, and TGF-beta signaling, epithelial-mesenchymal transition, myogenesis coming from the MyCAF, cancer-associated fibroblasts, and the desert tumor are enriched in Wnt beta-catenin and HOX signaling. So because those pathways are usual suspects that we find in clinical samples across immune phenotype, we compare this pathway enrichment analysis and gene set enrichment analysis with our clinical samples. So for that, we have leveraged 3 clinical trials, phase 2 and phase 3, from ovarian, bladder, and lung cancer. And you can see here that between the mouse stump model and the archival human tumor, the same pathway are highlighted, similar pathway are highlighted across immune phenotype. So which give us confidence that this immune phenotype are clinically relevant for the next step of our investigation. So we still didn't solve our question, what is controlling this heterogeneity of T-cell distribution among tumors of the same array? So we have asked 3 different questions. Tumor cells could be different. I mentioned to you that at seeding, those tumor cells are identical, but what happened after a few weeks, those tumors could diverge over time. The second point we have addressed is maybe the quality of T cells is different. So some tumor nodule could recruit antigen-specific T cells and other more bystander cells in terms of repertoire. So we went back and looked at the T cell infiltrate. And the last step is the tumor tissue microenvironment. So maybe the T cells that are seeding in those different nodules, we see different myeloid cells and stroma populations that could shape their function over time. So if we start to tackle the first question, we have implanted different cell lines in the stump tumor array. And what was intriguing to us is we always observe this local heterogeneity of tumor immune phenotype. If we implant a B16F10, a melanoma cell line, a PDAC, KPP cell lines, or a GEM-derived cell line from non-small cell lung cancer, in this stump array, we see that we obtain different proportion of tumor immune phenotype, and this is the number of tumors that are being analyzed. And for example, melanoma is enriched in immune cold tumor, and non-small cell lung cancer enriched in excluded tumor. So that tells us that tumor cells influence the probability for the tumor of developing a certain tumor immune phenotype, but they are not strictly deterministic. Something else is locally also controlling the development of a tumor immune phenotype. We also have performed whole exome sequencing at 10 days or 15 days down the line, and to compare the accumulation of mutation across a tumor nodule, and we don't see any significant differences there. So that tells us that probably the tumor cells give a probability to develop a certain tumor immune phenotype, but something else is controlling this local heterogeneity. So the second question we're asking are T cells recruited in those different nodules different in terms of function and repertoire? So for that, we went back to our punch biopsy and we performed single-cell RNA-seq of those TD tomato cells. And what we have seen is that based on a relative and absolute abundance of the different T cell subsets, we do not observe a difference between rejected tumor, efficient rejection, inflammation, and excluded tumors that are a failed rejection. We have the same proportion of T cells. So once we look at that, we looked at early time points. So this is at day 8. And T cells start infiltrating the tumor around day 6. So day 8 is already the first time when we can really distinguish tumor immune phenotype. And we wanted to look very early to see what's happening at early time point. We're now looking at later time points because what those T cells are going to become over time might change depending on the TME they are seeded into. So no difference in in relative abundance of the different T cell subtype. So after that, we looked at the repertoire of T cells, and we show that different tumor nodules shared a similar T cell repertoire between rejected, excluded, and inflamed tumor. So again, this data seems to be the driver of this different behavior of the tumor nodule. However, the advantage of the system is that we can now, because those tumors are implanted in the same animal, we can now look at the same clonotype of T cells that is seeding two different nodules. So the T cells with the same repertoire that are seeding an inflamed, coming from the same blood, an inflamed tumor versus an excluded tumor, and perform a differential gene expression analysis. And then what we see is that T cells that are seeding an inflamed tumor microenvironment show an increased translation and mitochondrial biogenesis associated gene signature. That would give us a hint that maybe those T cells are functioning better in an inflamed tumor microenvironment. And this is a question we are asking now. So if I need to summarize what we just saw, we saw that we need to recruit antigen-specific T cells to reject those tumors. However, those T cells need to be spatially distributed in a certain in an inflamed tumor in the core of the tumor. But now the question we are asking is good to have the T cells, is good to have them recruited, but what about their function, right? So relative abundance or absolute abundance is not enough to distinguish a tumor outcome. So to look at their function, what we are doing is perform different type of imaging. So we're doing epifluorescence imaging on this tumor array at low resolution. You can see the whole array, but we also zoom into each tumor nodule and perform 2-photon imaging to look at T cells and tumor cell interaction. And we can now look at T cell dynamic over time. You can see here the tumor, the collagen, and the blood vessels. And you can clearly see the different pattern of migration of the T cells. So we can follow migration and recruitment from blood, but what we were mostly interested is to be able to quantify T cell function in the tumor microenvironment. So for that, we have developed a calcium sensor. So those tumor cells that are expressing the BFP at baseline are going to express the calcium sensor GCAM6, which is going to turn green fluorescence when the concentration in calcium, intracellular concentration in calcium, is going up. So each time a T cells here with TD tomato is poking a hole in the plasma membrane of the tumor cells, there is an influx of calcium from the extracellular space to the intracellular space, and now the tumor cells turn green for a few minutes. So what we are doing is to translate the number of calcium flashes and the duration of those flashes as what we call a calcium flashing index, and translate that as number of tumor attack. So if we implant those tumor cells in vivo, in stump, what we see is that immune cold tumor that have no infiltration in T cells have a little green flashes. At the opposite, when those tumors are highly infiltrated by T cells and actively attacked, here is a dual engager, TDB, to force the attack of tumor cells by T cells as a positive control, you can clearly see there is a lot of green flashes in the tumor cell. However, even locally, you can see some heterogeneity with a very strong pocket of functional T cells and some places where the tumor are flashing less, also they are infiltrated. So it tells us that being able to read out the function of T cells in the TME is something that is going to be important, because only presence of T cells doesn't tell the whole story. So, and then I mentioned we quantify what we call a tumor flashing index, integrating the number of flashes and the duration of flashes. So what we learned with that system is that if we compare inflamed and excluded tumor, the total abundance of T cells is similar between both tumor type, and even sometimes we find more T cells in the excluded than in the inflamed tumor. But if now we look at the flashing index, you can clearly see that those T cells seem to be functioning better in inflamed tumors than in excluded tumor. And if we correlate that with outcome and rejection of progression of tumor, we clearly see that the inflamed tumor are associated with a higher rejection rate than excluded and desert tumor. So that tells us that those T cells with the same quality and the same repertoire that are seeding two different nodules, inflamed or excluded, seems to have their function better supported in an inflamed tumor microenvironment. So this brings us to look at the tumor microenvironment, but more particularly the myeloid and the stromal circuits. So for that, we came back to exploring the contribution of myeloid and stromal cells by leveraging single-cell RNA-seq. So we performed punch biopsy, and this time we subcluster the myeloid cells and the stromal cells across tumor immune phenotype. So what did we learn looking at that? So first of all, if we classify the tumor from rejected to inflamed, excluded, and desert outcome, we can clearly see that the inflamed and the rejected tumor has an enrichment in these orange cells that are monocyte-derived dendritic cells, and monocyte-derived cells. So those cells in general have a stimulatory phenotype with some ISGs and also enriched in dendritic cells that are expressing co-stimulatory molecule or maturation markers. At the opposite, in the cold tumor, we see okay, I think I lost you. At the opposite, in the cold tumor, we see an enrichment in immunosuppressive macrophages here in violet. And we also see, if we look at the neutrophils, those neutrophils are excluded in inflamed and rejected tumor, but we don't see any recruitment in desert tumor. And if we subcluster a bit more, we see that we have an enrichment here in the cold tumor in TREM2 positive macrophages, some type of Langerhans cells, and a subtype of dendritic cells type 1 that seems to have a different phenotype than the other immune phenotype of tumor microenvironment. And if we now look at the fibroblast, we can clearly see that the immune-rejected and immune-inflamed tumor are enriched in chemokine-producing fibroblasts. So we define those fibroblasts are producing many different chemokines, but we do not call them ICAF or inflamed cancer-associated fibroblasts because we do not see any production of pro-inflammatory cytokine. So we mostly see chemokine production. So this could be two reasons. All those tumors are very early and those fibroblasts are still on the way of differentiating toward maybe inflammatory CAF or maybe that's a different subtype of fibroblasts that still didn't see these chronic inflammatory settings for long. So, and then what you can see is that inflamed and rejected tumor are enriched in chemokine-producing fibroblasts in green, but in the opposite, desert and excluded tumor are enriched in immunosuppressive fibroblasts. So the excluded tumor, you have this expansion of this myocaf, TGF-beta, LAR15 positive. In the desert tumor, we see two different subtypes of cancer-associated fibroblasts that also seems to be suppressive based on their gene signature. But we are now working at looking into human tumor to see if those populations exist also in human tumor. So what we realize quickly is that different subtype of myeloid and stromal cells that seems to talk to each other based on receptor-ligand interaction seems to populate the different tumor microenvironment locally in this tumor array. So if we looked at what those cells are producing, the monocyte and monocyte-derived cells seems to be the higher producer of T cells chemoattractant, CXL10 and CXL9, together with the endothelium. And if we perform a receptor-ligand interaction analysis, we realize that the chemokine-producing fibroblasts produce them on their side the myeloid chemoattractant, when on the other side the myeloid cells express the receptor for those chemoattractants. And so that tells us that maybe those cells have different function, one being more specialized in T cell recruitment and the other one being more important for myeloid recruitment. And so this is only a correlation, right? Looking at the abundance of those different cell types and the expression of chemoattractant based on single cell RNA-seq. But if now we want to have a correlation analysis, that means intervene on these monocyte and myeloid cells and stromal population and see how that control T cell recruitment in the early events of developing a tumor immune phenotype. So we have developed different technology. I mean, we have used a different technique to deplete neutrophil, monocyte, or both of them in the early time of T cell of tumor development. And what we can say is that if we deplete monocyte, neutrophil, or both of them, we basically shift tumor toward a more desert phenotype. So this is looking at T cell distribution. Now there is less T cell recruitment, and this is associated with less tumor rejection. So it seems that those myeloid cells are important somehow to attract T cells and to maintain them at the tumor site. If we now deplete the fibroblast and we use a new mouse model that had been developed in the Turley lab, where we can use a DPT Cray mice crossed with a DTR lock-stock lock system, where we inject diphtheria toxin and able to deplete the whole population of fibroblasts in the skin at tumor implantation. We also shift the tumor immune phenotype toward a more desert environment. So this is based on T cells. If you look here, the tumor are in green and the T cells are in red. Once you deplete the fibroblast, you have way less infiltration of T cells in tumor. If you look at the myeloid cells, once you deplete the fibroblast, you also decrease the recruitment of conventional dendritic cells and monocyte-derived cells. So it seems that the fibroblast depletion prevents myeloid and T cell recruitment and shift tumor toward the desert phenotype. And we really focus on those early events when the immune phenotype develop in vivo. So if we summarize that in a model, we think that like so at the implantation of the tumor array, skin fibroblasts are perturbed and can develop toward two different trajectories. One is developing toward the chemokine producing fibroblast that will recruit a lot of chemoattractant for myeloid cells and recruit monocyte and dendritic cells that are going to secrete a lot of lymphoid attractant to attract effector cells and then lead to tumor rejection. At the opposite, the other trajectory is this fibroblasts become a TGF-beta calf. They will induce more an excluded phenotype and a failed rejection. So what we have done afterward is to look at cancer immunotherapy treatment and dynamic in the tumor microenvironment of immune cells migration and immune phenotype over time. So we took the example of treated with a combination of anti-TGF-beta, anti-PD-L1 in the stump tumor array. And we have looked at 4 different arms: control mice, mice treated with single-agent anti-TGF-beta, mice treated with anti-PD-L1, and mice treated with a combination therapy of anti-PD-L1 and anti-TGF-beta. And we looked here up to 15 days of total T cell abundance in those different tumors. So you see here the number of tumors that we have analyzed, and one dot here is one tumor. So if we look at the total abundance of T cells, you can see that both anti-PD-L1 and the combination therapy, anti-PD-L1, anti-TGF-beta, inducing a recruitment of T cell over time. And this is this is aligned with the result presented by different groups and published by different groups in the past, including the groups at Genentech in our departments. But what is surprising also, both anti-PD-L1 alone and combination treatment is recruiting those T cells. Only the combination treatment anti-PD-L1, anti-TGF-beta is effectively rejecting tumor over time. So that means that recruiting T cells is not enough to induce tumor rejection. So we wanted to look at how immune phenotype dynamics happen over time in those tumors to understand what's happening here when those T cells are recruited at the moment of recruitment that seems to control tumor outcome. So for that, we went back to our stump image analyzer app, and we represented what we call heat maps. So we looked at the lifetime of a tumor. So one line is one tumor trajectory. Observed here it's a 30 days scale. During 30 days, we image those 1,200 tumors, and we can represent different parameters along this tumor trajectory. So the tumor growth rate here, when the tumor, there is green color, the tumor is growing. When it's blue, the tumor is regressing. Then we looked at T cell abundance. When these lines are very yellow, you see a lot of T cell abundance, total T cell abundance. When it's dark, those tumors are very cold. And then the last parameter we can represent over time is the tumor immune phenotype. So we see that some tumors start as an inflamed tumor from the get-go and stay inflamed their whole lives. This is the green here. And then we observe some transition. Some tumors start excluded, become inflamed. Some tumors stay desert. Some tumors transition back and forth between an inflamed, a desert, an excluded, an inflamed. So what is important to us is that tumor immune phenotype are not stable over time. They seem to transition back and forth between a different spatial distribution of T cells between inflamed, excluded, and desert tumor. And if we perform unsupervised clustering, we can clearly see that some tumor trajectory cluster together, the responder cluster together, and this is leading to tumor rejection, and the non-responder cluster together. So we looked at different ways to represent the transition in between tumor immune phenotype during treatment. And what we have done is representing, so we have performed an unsupervised clustering, try to see if there are different patterns of tumor behavior. And what we see is those gray lines represent a different tumor trajectory over time from day 6 to day 18. And this is the number of tumor trajectory per class that has been analyzed. And this colorful line is the average of those 80 tumor or 75 tumor, 49 tumor. And we represent the total T cell abundance. That means once tumors are low here, they are more cold. When they are high, they are recruited a lot of T cells. And what we call the infiltration ratio, where those T cells are located. When they are on the left here, they are rather outside the tumor. When they are on the right, close to 1, they are really in the core of the tumor. And the software tells us that there is 3 different behavior in tumor trajectory. Some tumors that start as desert and then become highly excluded, that means recruit a lot of T cells, but those T cells are stuck outside. So some tumors that are starting as desert and then progressing quickly to an inflamed phenotype, so recruit a lot of T cells that are infiltrating the tumor core. And then tumors that start desert and become excluded, so T cells hang around for a long time around the tumor, and then they become inflamed later in time. And if you look at how outcome is correlated with those 3 classes of tumor, only 1 class of tumor is getting efficiently rejected, is the class number 2. So that means you need to recruit T cells, a lot of T cells, but those T cells have to be able to seed an inflamed environment and get into the tumor core very quickly. If they start being excluded for too long, they might become dysfunctional, and then even if later in time they manage to infiltrate the core, this doesn't lead to an efficient rejection. So that tells us that looking at dynamic of spatial distribution of T cell in time is important to predict rejection, but also it seems to tell us that the tumor microenvironment keeps a memory. So there's kind of an imprinting. When those T cells sit too long in a dysfunctional TME, then it's difficult to reverse their function later in time. So that tells us that if we think about the patient, maybe that's the reason why an inflamed tumor at start of treatment, once we looked at immunohistochemistry, is associated with responses because the new T cells that you are priming or generating with immunotherapy or checkpoint blockade are going to see the inflamed microenvironment, and you have this window of time where their function will be supported in the TME. However, what will happen 3 months down the line or what happened before, we have no idea of that, right? So if we summarize all of that in a scheme, it seems that systemic immunity and it's very important. We need to prime antigen-specific T cells that are going to circulate in blood, but is not sufficient to induce tumor rejection. Once those T cells see the tumor microenvironment, they are going to meet different myeloid and fibroblast units that are talking to each other. And if this tumor microenvironment is enriched in chemokine-producing fibroblast and monocyte-derived cells with a high T cell abundance and an early conversion to an inflamed phenotype, the tumor is going to move toward a regression state. At the opposite, if we have more TGF-beta CAF, low monocyte and dendritic cell content, low T cell abundance, and excluded or desert phenotype, or even a conversion to an inflamed phenotype that is too late in time, then the tumor will progress. And the immunotherapy combination seems to move this. So the question we're asking is this immunotherapy combination moving the tumor toward a more regression state? So at the end, this is important to realize that the tumor microenvironment dictates outcome in those tumors. And I think I jumped a slide, that's why I was confused. So if we look at those tumor trajectory by treatment, if you see like in the control group, the tumors stay rather in the cold quadrant, bottom left. Anti-TGF-beta doesn't do anything by itself. The tumor also hangs around in the bottom left quadrant. Anti-PD-L1 in this tumor model doesn't show efficacy by itself because it's recruiting a lot of T cells, but those T cells stay excluded. And then the combination treatment, anti-TGF-beta, anti-PD-L1, moves the tumor toward an inflamed phenotype. And this is what is represented here, that immunotherapy is pushing those trajectory toward more regression. So this is in mouse, and all of that has been done using a tumor array. So we ask, does that transition happen also in patient? And the answer is yes, by looking at immunohistochemistry before and after checkpoint blockade. We have transition of immune phenotype from desert to excluded, but also excluded to desert. So both directions are happening. And because we can argue that immunohistochemistry, you're looking at 2 different time points, 2 different resection or biopsy, and you don't really look at the same cells. So we are also leverage a collaboration that Genentech has with a group in Netherlands, De Vries lab, where they really looked at anti-CD8 PET imaging tracer to look at infiltration of tumor before and after checkpoint blockade. And you can see here it's a urothelial cancer. In orange, the T cells are really clearly infiltrating the nodules after checkpoint treatment. However, there is no rules yet of which type of infiltration is associated with outcome. And again, we wanted to ask, is it possible to have different tumor immune phenotype inside the same patient with different tumor lesion? And this is a case of triple negative breast cancer with a primary tumor here, a pleural metastasis here, and different lung mets. And you clearly see that the primary is inflamed, is recruiting T cells, is hot nodules, but the lung nodules are pretty cold. So there is also this heterogeneity inside the same patient and for tumor immune phenotype across multiple lesions. And what we are doing now is to go beyond CD8 T cells and perform longitudinal imaging of other TME components. So everybody is focusing on CD8 T cell distribution, but can we use the distribution of other cells in the tumor microenvironment to better predict outcome in tumor? So we look at fibroblasts, neurons, blood vessels, dendritic cell macrophages. We also look, we made some reporter for different elements of the tumor microenvironment, one of them being the hypoxia, and we try to integrate all of this information in a way that will make a more complex pathology map of the TME. For example, can we expand the definition of immune phenotype to myeloid cells? So if we look at CD11C positive cells in those tumor nodules, we can see that some CD11C cells are infiltrated in the core, other tumors, they have them excluded. And we are also not only looking at those different cell types, but we are also performing spatial transcriptomic of those different nodules, looking at inflamed tumor or excluded tumor for both dendritic cells here and T cells over time. And we also perform laser capture of those different tumor nodules, classifying them by different immune phenotype for myeloid and T cells, distribution as excluded, inflamed, and looked at by proteomic, look at the different chemokine, extracellular matrix deposit in those different tumor nodules. So going beyond cell types, but also looking at soluble clue and extracellular matrix. And the future direction, we try to integrate all of this information together, combining not only high content imaging, tumor growth outcome, immune cell migration, but also spatial, transcriptomic, mass spec, single cell RNA-seq. And we are now integrating some perturbation where we do some CRISPR knockout or overexpression in tumor cells and see how we change the tumor microenvironment. The idea is to really integrate this information together in a multidimensional space where we represent tumor in a t-SNE snap or spade trees, like now one dot here is one tumor, and this is representing 11,000 tumor in one experiment. And trying to understand which pathways are really critical to control tumor outcome and tumor immune phenotype development. Comparing that with the human tumor and see which pathway seems to be translating from mouse to human. And I will go quickly through the last piece of information that we have been working on. As I mentioned previously, we're really focusing in our lab of inflaming immune cold tumor. Those immune desert tumor have the highest unmet medical needs. If we looked at incidence, there's still a big piece of the pie. There is many indications where those tumors have high prevalence. And we think that it's important to tailor immunotherapeutics to this kind of cold tumor. And what is known, what the field is at the moment, if you look at those different, I invite you to read those review about updated cancer immunity cycle and which pathway are enriching this different tumor immune phenotype. So we know the inflamed tumor have an enrichment of exhausted T cells, chronic antigen stimulation, Tregs. So everything that would be checkpoint blockade, Treg depletion would be highly relevant for inflamed tumor. Excluded tumor are enriched in fibroblast extracellular matrix, so all therapeutics leveraging fibroblast biology would be relevant for excluded tumor. If we focus on the desert tumor, basically those tumors for long term are described as a lack of something. So they are more ignored, lack of antigen presentation, MHC expression. There are also a lack of myeloid cell infiltration, more particularly dendritic cells. So we have been looking into it, how to leverage different therapeutic angles to inflame those tumors. And we mostly focus on the lack of stimulatory dendritic cells. If we focus on the lack of stimulatory dendritic cells in those tumors, and the work that has been done in the past by multiple labs that have been working on FLT3 ligand, we wanted to first give a good oversight of how immune cold tumor, what is the correlation between infiltration of T cell and infiltration of dendritic cells across the whole TCGA. So what we have done is based on bulk RNA-seq and looking at gene signature, we classify the tumor as cold tumor or hot tumor across all those and different indications. And now we looked at DC gene signature to look at the abundance of dendritic cells between cold and hot tumor. And we clearly see that across indication is highly significant. When there are T cells in the tumor, there are disease, and when there is no T cells, there is no disease. So maybe I should say the other way around, which is when DC are there, T cells seems to be maintained in the tumor. When they are not there, they do not seem to be maintained. So the hypothesis was that like a low number of dendritic cells in tumor is a limiting factor for cancer immunotherapy, and expanding and maturing those dendritic cells would enhance response to checkpoint blockade across indication. And that would be more particularly relevant for immune desert tumor. So we have developed a pipeline that allows to expand dendritic cells with FLT3 ligand, and then load those tumor with load those dendritic cells with tumor material, combining radiotherapy or chemotherapy, maturing those dendritic cells with multiple adjuvants, and there are many options there, and then develop PET bioimaging probe to track their dendritic cell expansion over time in vivo. And I will only give you a little bit on the first step here of what has been developed at Genentech to expand those dendritic cells. And this is based on the work of many groups in the past, more particularly at Sinai by Miriam Mera's lab, but also Josh Brody and Sandra De Maria. And we develop new clinical molecules that is made of a fusion of the growth factor domain of the human FLT3 ligand from position 27 to 167. This is this extracellular domain. And we put that in fusion with a native spacer, a native ligand here, spacer, together with the Fc fusion that is effectorless, that we call NG2LH. This effectorless fusion is a fusion of IgG1 and IgG2, and it also carries the N297G aglycosylation mutation to be completely effectorless. So if you look at binding of different Fc gamma receptor, it's really dragging down to very low binding. And then once we look into two different assays of ADCC and ADCP and compare that to all different type of format of effectorless, such as NG2LH, N297G mutation, and then IgG4, you can clearly see that ADCC is very low, but also this time with this new format, ADCP is also inexistent. And in comparison to, for example, IgG4, or N297G here, that still conserves some ADCP. So we really wanted to expand the dendritic cells, but we did not want to trigger any ADCC or ADCP for efficacy reason, but also for safety reason. So this Fc fusion, the goal was also to improve the PK property. If we inject in mice, the growth factor is the half-life is going to be pretty short. Now with this Fc fusion, the half-life of the molecule stay longer in circulation. And once we have done a cyno experiment, we also see that we can control DC expansion. It's not because this PK is extended that the expansion is completely out of control. If we inject a small dose of a molecule, you see a peak of expansions that contract very quickly thanks to TMTD, target-mediated drug disposition. That means when the cell expand, they are going to sink the drug that is in circulation and decreasing exposure of the animal, and then the cells will contract. If we inject a higher dose, now we're going to maintain this expansion over time. So basically, by injecting a certain dose, we can predict the fold expansion and the duration of expansion. If we look in tumor, what we look at is this is untreated mice, and then FLT3 ligand growth factor simple with short PK, and this is with long PK. We clearly see by day 9 here, when the blue molecule is gone, the dendritic cell not expanded in tumor, but with the FLT3 FC, the dendritic cell type 1, type 2 PDCs are nicely expanded in tumor. The monocytes are expanded in blood, but not in tumor in mice. And because FLT3 ligand as a cancer treatment will always be part of a combination therapy, FLT3 alone will not be used in clinic. We think that we need to combine with some debulking agent, maturation, and checkpoint blockade to reinvigorate CD8 T cell responses. We have used that in a very cold tumor model, at least in our facility. The B16F10 model is very high bar, it doesn't respond to any immunotherapy in our hands. And what we have done is treating those tumors with FLT3 ligand, poly-IC, and anti-PD-L1 with increasing doses of FLT3 ligand. We see that we have a better efficacy with increasing doses of FLT3 ligand. And we have a dose-dependent efficacy in the combination settings. And even if we inject multiple times now with multiple cycles, we can improve even more the combo efficacy with 2 injections of FLT3 ligand and really delay this rebound of tumor growth. And then the last piece of information I wanted to give you before we get to the question is showing a bit of data from the phase 1A clinical trial in healthy volunteer when we have done a single ascending dose of FLT3 FC and 2 courses of multiple ascending dose, 2 injections. And what the prediction was telling us, has been published, that the PK/PD modeling tells us is that with multiple dosing every 3 weeks, so 21 days, that should be the PK profile of low doses and high doses of therapeutics, where if we look at the PD here, low doses, you expect to have an oscillation in myeloid cell expansion with a contraction, so the cell should go back to baseline. But if you now inject like a big dose regularly, you're basically going to maintain this DC expansion for a very long time. And that's what we are seeing in the clinic. When a single ascending dose from low dose to high dose, you see low dose small expansion and quickly contraction, but a higher dose, you maintain this expansion longer. And then in the multiple injection, you can see like at low dose, you have this oscillation of the different population of myeloid cells, but at high dose, you maintain this expansion. And this is a summary of the dendritic cell and monocyte expansion across species in mouse, cyno, and human, with a big fold expansion in blood that is higher, around 1,000 expansion in blood of mouse and cyno, that translates in a 50 to 100 time expansion in tumor. However, in human, we have a lower fold expansion, and this is known with, in the past with other FLT3 ligand molecule injected in patients. The fold expansion is lower, but again, we see this nice expansion of DC1, DC2, and PDCs. And in tissue, we have looked at infiltrate of those myeloid cells in liver, but in the small intestine. Also, you see those infiltration of myeloid cells at the peak of expansion. And then I just encourage you to read the updated cancer immunity cycle incorporating the tumor microenvironment in the big picture here, more particularly the TLSs for inflamed tumor, all the MyCAF biology for the excluded, and some more toxic environment for the desert tumor. And as I mentioned, it's a big teamwork. A lot of people have been involved in my lab. And that's a wonderful team. They are really done a super good job. I'm so glad to work with them, and a huge team for the FLT3 ligand team, because this takes a big group effort to bring a molecule to the clinic. Thank you so much for your attention, and I'm happy to take questions.
Outstanding, and congratulations on this great work. If you have questions, you can put them in the chat, or you can unmute your microphone and ask. I'll start off. I'm particularly interested in the dendritic cell data that you showed, and I was wondering if you all have characterized those CDC2 populations. Are they CD16 positive, CD16.2 positive DCs that you're seeing be expanded in some of the data that you showed?
Yeah, so I think, so the manuscript, I have to be careful of what I can say and not say yet, because manuscript is not public.
Okay, understood.
No, but we have looked at the different type of monocyte expansion, the different type of DC1, DC2, PDCs. So all those populations are expanded with different ratio, and the DC2 being CD16 negative, but we also have the CD16 positive cells that are also expanded. So probably with different proportion of expansion. That's what I can say.
Great. Christopher Garris had his hand up first. Christopher?
Oh, that was a fantastic talk. I had a question about the fibroblasts. Do these fibroblasts have similar phenotypes to lymph node fibroblasts? And if so, or TLS fibroblasts? And are there any potential ways to maybe induce that, like with lymphoid tissue inducers or something of that ilk?
Yeah, so I think it's a very hot topic at the moment. So if you look at those single cell, I mean, the thing is the tumor are very small and very early, right? So we also know that for TLSs, you need certain time to develop TLSs in mouse, and mouse are not really good model to develop TLSs. So I would say that probably at the time we are looking at right now, which is day 10, you know, I cannot say that those are FRC-like fibroblasts that would require a sustained signaling through lymphotoxin, for example. We see that there might be might be like on a direction toward it, but I don't think we can say that we have FRC-like fibroblasts in this model just because this is very early tumor. However, I know that across the pharma industry, many people are trying to induce TLSs and are working on reprogramming the tumor microenvironment to produce lymphoid chemokines, CCL19, CCL21, CCL13, to induce this TLS development. And I know some people are getting successful with it, so I think it's doable. However, yeah, so two things is doable. However, mouse is a challenging model to do that, and it's doable, but you need to look at longer time. And I'm really curious to see where the field is going to go with that, because I'm sure we're going to see some development in the future.
Johanna, Joyce?
Hi, Christine. That was an absolutely fantastic talk. Thank you so much. I just loved the nature study, the tumor microenvironment study. It was just it's so powerful. I have so many questions. I tried to limit myself to one. It's about the comment that you made about T cells being trapped in scars, because that's very similar to something that we just reported in the context of GBM. So I just wonder if that's something that you could elaborate on a little bit more, if that's something, if you try to target those specific T cells, if so, whether that was effective or not. Yeah.
Yeah, I don't think we have done that, so I don't have a lot to answer. I mean, there's so many things we can do with the system, but we are a small group, so we cannot do everything. We're happy to collaborate. So yes, we see those T cells staying for a very long time. We have not really, I cannot say we have leveraged them. What we would like to do is really to biopsy them and try to understand from them, because if they are there, that means they have been efficiently rejected tumors. So maybe can we learn from them their phenotype, but also if we do some perturbation in the T cell compartment, if you, for example, think of a small CRISPR screen or T cells, can you biopsy those tumor nodules that have efficiently rejected tumor and look at perturbation that is making those supercharged T cells that are able to reject tumor? That could be an option and what we would like to do, but I don't have a lot of information about those T cells just because we didn't really characterize them properly. Maybe we can reach out to you.
Yeah, no, that would be great to talk about this. Yeah, thank you. Thanks again.
And I have another, sorry, I wanted to add something a bit.
Go ahead.
We also see sometimes in some tumor model, some tumor cells that are seeding, that not developing tumor immediately, but that have like those isolated cells that stay there dormant for a long time. And we then sometimes few months later, they start emerging and making tumors. So we forget about those mice and the vet call us, oh, you have tumor like a few months later. That would also be interesting to see how they interact with some resident population.
That's exactly where we see them, is in the context of long-term dormancy. So yeah. I'm going to write to you afterwards and we can chat more offline. Thanks.
Perfect. Thank you.
Daria, you have a question?
Wonderful talk. I was wondering, FLT3 ligand is known to play a significant role in expansion of Tregs. So I was wondering if you ever looked at the Tregs in your model.
Yes, so we have looked in depth. We have done in-depth immune phenotyping of the T cell population too. I didn't present it. It's correct, and this has been shown by others in the past, that when you treat with FLT3 ligand single agents, that's a study done in Australia in melanoma patients, where basically you see expansion of myeloid cells, and if you continue expanding around day 15, so the peak of dendritic cell expansion will be around day 8, 10, and then you will see a peak of Treg expansion around day 15. So it's correct that in single agent you will expand Tregs. However, if you combine that with an adjuvant now, you will shift your DC from more regulatory to more stimulatory, and then you will invert you will always see Treg expansion as a feedback loop of priming, right? But you will really shift toward a higher T effector on Treg ratio. So you will expand more CD8 effector cells than Treg expansion on a ratio basis. So I think that's probably the shortest answer I can give you. Don't use FLT3 ligand alone. Yeah, for sure.
Great. Abi?
Yeah, it's a great talk. I have a couple of questions on the CAF population, like especially whether the surface phenotypes are characterized like any kind of, like a PDL1 or TIM3, TIM4, those kind of expressions, are they upregulated? And number two is the dynamics after immunotherapy, like say inflamed to desert or desert to inflamed, those kind of switches are happening. So do you think, does it correlate with your adaptive resistance, which is one of the key factor right now in immunotherapy, and which is the limitation? Can you highlight on those two topics? Thank you.
Yeah, that's a very good question. I will be disappointed because I don't think I have a lot of answer for you. But we have not really looked at surface protein expression on those fibroblasts. We can probably deduce things from the single cell, but we have not looked at checkpoint on the cell surface. We still, the highest characterization we have done are very early time point. I mentioned to you now we are doing late time point, and that's also an important piece of information when you want to look at all those markers. But by day 8 and 10 is still a bit early. So I can probably get you answer from the single cell. I don't have any stainings done on those cells, sadly. And then the transition of immune phenotype with time. Yes, so I think clearly once you start being in the day 20, day 30, what you see that tumors that have been infiltrated, they go through phases of like growth and then contraction and then regrowth and then contraction. So clearly we're looking into resistant mechanism here, more particularly after immunotherapy treatment. And if you biopsy before and after, you could look into mechanism of resistance. We have not done it yet. So that's also something we should be doing.
Thank you.
Dr. Rosandra Kaplan. Hey, Rosie. Yeah, you can hand up.
Hi, how are you? I have to say, I absolutely love hearing you speak, Christine. It was just terrific. And as Johanna said, I have a ton of questions, but I'm gonna really limit it to one because there were so many aspects. It was interesting. I love the early myeloid stromal cell crosstalk focus. I feel like I've been shouting that from the rooftops a long time, and it's but it's hard to get concrete data on that, and I feel like you do. So my question is, the beautiful trajectories you show of the different microenvironments and how they all become, even if they become inflamed, how they get there matters, and this kind of microenvironmental memory you talked about. Do you think therapeutically, is there a way to intervene that would change that? Like if a patient was to get, let's say, chemotherapy when they were in the immune desert state versus if they were in the different state, would that change the trajectory one, and could that restart it? And do you think you could ever get to the point, even though you had to go through all the steps and you get inflamed and you don't get the same effect, could you like retrain them? Could it be reversible or reprogrammable?
So I'm not a medical doctor, so big disclaimer. I cannot recommend anything on like treatment. I'm just a PhD, and so based on mice, I would say
Not just anything. Don't say just.
Based on like, okay, if our theory is true, the issue with if you don't have dendritic cell in a cold tumor and you are going to perform chemotherapy, this is going to be challenging, right? Because you don't have anything to load your material on. So that's maybe where you really need to leverage both aspects to have like increasing. There's also two types of chemotherapy. If you have an immunogenic chemotherapy, then you can release some inflammatory signal at the same time and then recruit more cells and then allow priming. Maybe you can get there. I'm not sure. We have not done that chemotherapy experiment, and we have done chemotherapy experiment, but the problem is there are so many chemotherapy with different mode of action That is also a big bucket. We have tried to detangle that, try different type of chemotherapy combination with FLT3 ligand, for example. I don't have a quick answer. This has been challenging discussing with the clinical team because also it doesn't translate mouse human. So
This
I have
is a
good
huge
argument to say
challenge.
yes, and I have good argument to say no. I'm not sure it's helping the.
Yeah, I would love to continue the dialogue though, because I think as we move forward with microenvironmental modulation, we're going to need to think about combinations. And I'm not sure the mouse is an easy way to do that when we're thinking, you know, the patients they're usually going to get chemotherapy or have gotten, or they'll get it again if we're talking about multiply relapse. Anyway, awesome. Love it. Thank you.
We also have seen that we have treated mice tumor with radiotherapy and with chemotherapy, and the dynamic, the moment the tumor starts regressing is very quick in mice, but in humans, sometimes you have to wait like several weeks, you know. So I think the timing is also very challenging of those combinations. It's not only we put agent 1, 2, and 3 together, it's which timing. And we also know that checkpoint and radiation, for example, if you do one before the other one, is going to show
Totally
synergy.
different effect.
So the timing is going to be something important too. And yeah, that's why those combinations are pretty challenging in clinical practices, I think.
Yeah, that's great. Thank you so much.
Dimitri?
Yes, hi. I hope we have time. I have a relatively short question, but I'm not sure whether you have an answer, because you have extensive studies, very early events, and which is incredibly important. But I couldn't resist to come to these questions. Do you know what happens? I mean, you have a chance to look at microenvironment when the tumor becomes a little bit bigger. The changes in microenvironment become very different. For instance, neutrophils, what you just described, has been now very clearly implicated as anti-tumor agent, but only and only if the tumor either small or already rejected by checkpoint. As soon as the tumor becomes a little bit bigger and growing, their functionality changed dramatically. I think the same with monocytes. It might be in other cells as well. I'm pretty sure something's published. So my question is just, how do you think that concept of this shifting desert with inflammatory will hold up when tumors become a little bit more clinically relevant?
Yeah, this is a very good point, and I completely agree with you that early events and late events are different things. Okay, here we have like for very large, so what happened in those different nodules, right? You can see them very dense or far away from each other that you can follow tumor longer. Because what happens if you see them so quickly, they're gonna grow and they grow into each other. Sometimes if one is an inflamed, one is in cold, they keep this polarization of like a cold versus a hot together. However, when they start being very big, the imaging is not doable anymore because, you know, you have a lot of diffusion of lights and like start being like a necrotic. So all of that is difficult to follow by live imaging when you get to very big tumor. So probably your system is not well adapted for large tumor. However, we have done different treatment early versus late, and I agree with you that probably the conclusions are different than what I was mentioning, that we have started by understanding the system and establishment of the immune phenotype. That was one part. Now we're working on like intervention studies with very late tumor. But however, I think our system is not super well adapted for that, and that's one caveat. What we wanted to do with that is really to go beyond the in vitro model. So people do a lot of like spheroids, co-culture experiment, organoids, which is great in vitro. It's a good tool for high throughput and high throughput imaging, but you still don't have vasculature, innervation. So we wanted to bring that field in vivo and look at small tumor in vivo in more conventional environment with like ECM, fibroblast, vascularization. I don't think this would be the best model to look at bulky tumor. Also, you can get there. If you let it grow, it takes the whole year. But then maybe now with this super clearing agents that everybody's talking about, when you can make things more transparent, the transparent mouse will stay alive, maybe you can look at more bulky tumor with our system. But live imaging in bulky tumor doesn't get you very far in general.
Awesome. Christine, you have, I don't know if you're over a time limit, but there are 2 questions in the chat. The first one was asked by Camilla Bach. Is Camilla Bach still on? Do you want to ask your question or I can read it? So Camilla Bach.
Yes, I can do it. Thank
Go
you.
ahead.
So do you think that the big tumors implanted using a stem can recapitulate the same cellular phenotype and them dynamics as they would do if they were in their natural environment? And also, can stem be applied to other body parts of the animals or just the ear? Thank you.
So yes, we have tried stamp on different parts of the body. It works on the belly, it works on the back. You also have this heterogeneity. On the ear, you have like the spatial distribution of immune phenotype can be a little controlled by this centripete organization of the ear when the vascularization come in here from the base on the ear and then go do ramification like that. So then like the tumors that grow on the edge here are less well vascularized, so they are smaller. And bigger here because they are more like adipocyte-like. In the belly or the back, there is this heterogeneity is more homogeneous because the skin is more homogeneous. But you still see inflamed, excluded, and desert tumor. We have tried different cell lines, and we compare the different cell lines implanted in stump versus subcutaneous tumor. We have not implanted those tumors in other organs. What I can tell is that in general, they share the same infiltrate in subcutaneous and in stump. The only difference we have seen is for the melanoma cell lines, B16F10, when you implanted subcutaneous or on stump. On stump, it makes really small moles that you have on your skin. They stay small, they grow slower, melanoma, and they are more cold than subcutaneous tumor. And they metastasize pretty quickly to cervical too, lymph nodes. So there are differences. I think it really depends on your model, and you have to interrogate that. We are trying now to use that for a CRISPR screen or with more heterogeneous population of tumor cells. But yeah, I don't have an answer. That really depends on the model.
Okay, thank you very much.
You have a question from KJJ. It says, did you observe any toxicity related to the systemic delivery of the FLT3 IG1/2 molecule?
So, well, I don't think I can comment too much because this is not, I mean, I don't know if you're talking in mice or if you are talking in human. I can tell you that in the healthy volunteer phase 1, they look good. There is no much detected there. But this is going to be published soon, and I'm not MD, so I don't want to comment on that.
Understood. And then the final question is from Chanda. Is Chanda still on? If not, I'll ask it. It says a high CAF population leads to tumor regression, but does high CAFs also lead to desmoplasia and reduce contact of T cells to tumor cells? And then how does anti-PD-L1 and TGF-beta synergize for T cell infiltration and activation?
Yes, this is completely an important point. Like CAF by itself are a big family. So we have the chemokine producing CAF that we see in inflamed tumor, but in the excluded tumor, we have those TGF-beta CAF, and we see them very clearly that expand and that are expressing GLAR15. And then, yes, in that case, that's probably what's also part of the scheme of keeping T cell outside of the tumor nodule. We have not tested directly the depletion of those cells in our model, so I cannot answer that causality directly in the stump system. But yes, with anti-PD-L1 and anti-TGF-beta, we clearly see that those excluded tumors become inflamed, right? And that is clear. So those excluded tumors that have high TGF-beta CAF seems to transition toward more an inflamed phenotype. So yes, I completely agree with that comment there. That CAF are like two different populations, or even more, some good and some bad one. That the bad one would reduce T cell infiltration.
Awesome. Well, if there are no more questions, Christine, we, on behalf of the organizers of the Myeloid Network and our founders, we thank you for such a profound talk today. And we hope to see everyone here next month for our next speaker. And we will be sending updates and announcements to everyone soon. We are also on Twitter, so you can find us at the Myeloid Network on Twitter. Please follow our page. Thank you all. Enjoy the rest of your mornings if you're on the East Coast, West Coast, or afternoons.
Thank you. Thank you so much.
Thank you, Christian. Thank you, everybody. Fantastic.
Thank you.